Right. My name is Eric Admin with Sno-King Watershed Council, and welcome to Bacteriological Water Monitoring. So again, Sno-King Watershed Council is a local all-volunteer nonprofit, and we work for protecting local creeks. We're teaching a program that is part of a group called Global Water Watch, and Global Water Watch is a international organization that teaches people around the world to monitor local water supplies. This is an example of a bacteriological monitoring workshop that they were conducting in Kenya. And they've got programs going in different places, including Central America. So our agenda for this evening, we're going to talk about bacteria, about how generally they're beneficial, but also they can be dangerous, which is one of the reasons why we monitor them. We're going to talk about where bacteria in water comes from, and we're going to learn Global Water Watch techniques for bacteriological monitoring. So one of the questions we ask when we're monitoring water, is it safe to play, swim, fish, or drink? Now, generally, we're not drinking water out of our creeks around here, but we are playing in them, swimming in them, fishing in them. And so that's one of the questions we want to answer with our bacteriological monitoring. So just talking about bacteria, bacteria are extremely small, single-celled organisms. You can't see them. They're all over the place. They're in almost every environment. They're both inside of us as well as all over our skin. They're necessary for life. They play a necessary part in kind of how matter gets converted from one form to another. And this is kind of a little diagram that just shows what role they play. So they're kind of in that, they decompose things and help provide nutrition for other things. They're all kind of related in a way into different types of families. And so the families that we're primarily studying with this type of monitoring are what's called coliform bacteria. And in particular, the one we're concerned with is called E. coli. That's just one particular type of coliform bacteria. So E. coli is, when you find it in water, it doesn't absolutely mean for sure that there's fecal contamination, in other words, from human waste, but it's indicative of it. And so there's a close association between finding E. coli and fecal contamination, but it can come from a variety of different sources, whether it can be human or animal. And we'll talk about some of the different sources. That is a major cause of unsafe drinking water worldwide. In our country, we're pretty lucky that our drinking water is well protected. There's a lot of countries around the world. I've done some volunteer work in Haiti where people get their drinking water right out of the local river, and they don't have, they don't have sewage treatment like we do. And so you're really taking your chances drinking that water. So luckily we don't have to deal with that. And the particular strain of E. coli, which is pathogenic, is that one that's shown down there on the bottom of the screen. So it can come from a number of different places. Bacteria can get into water from a number of different places. One of the places is from pets. And so that's why there's been a renewed emphasis recently in the last few years on people picking up after their pet and not just leaving the dog poop on the lawn. Sorry. In fact, in Kirkland, they did a little pilot program where they put little flags out to mark dog poop that hadn't been picked up, because they were trying to incentivize people to pick up dog poop. And then they were out talking to dog owners, and this kind of highlighted how much dog poop wasn't being picked up. And so they were able to start reducing the amount of dog poop that was being left by kind of raising the attention to people about, hey, this is a source of water pollution, please pick it up. But they were estimating 6,000 pounds of dog poop a day was deposited in Kirkland parks, so quite a bit. And what happens is that runs off when it rains, that runs off across the land and then can get into the water. Another source of bacteria in water is in what are called side sewer failures. So the way that your house connects to the sewer system, if you're on a sewer, is via something called the side sewer, which is a pipe between your house and the main sewer. And in some cases, those are old, especially in houses that were built a long time ago, and they can crack. And if that happens, you might not be aware of it because it doesn't necessarily cause a problem for your house, but it could, depending on where that is, that could cause bacteria to get into the groundwater and into the local stream. So for instance, one of the groups we've been working with is called Thornton Creek Alliance, and they're in North Seattle. And they were having, Thornton Creek has high levels of bacteria, and some of the testing that they did discovered that there was one particular apartment complex that actually had its side sewer incorrectly connected to a storm sewer line. So they were basically getting sewage directly into the creek. So that was one source that they were dealing with. Animals in the stream is another source of bacteria, like livestock. So a lot of times for water quality purposes, people who have various farm animals are encouraged to fence them out so you don't have that source of bacteria in the water. And then locally, this is the UW-Bothell crow rookery. And there's just tens of thousands of crows that nest over there in North Creek at UW-Bothell. And they cause a fair amount of bacteriological contamination in the water themselves. Another thing that's associated with bacteria in the water is when you have high turbidity conditions. So this is an image of Little Swamp Creek in my yard with really turbid conditions during a high runoff. And just looking at the chart, we found super high bacteria levels on that particular time. So when you have a lot of dirt and just generally a lot of runoff in the creek, the dirt particles themselves can be attachment points for bacteria, but also that it's just associated with higher bacteria numbers. All right, we're going to talk about how to measure bacteria. So using Global Water Watch bacteriological techniques, they want you to do it the same way. So use the same techniques at the same location and roughly the same time. So the guidelines that they always give us is 10 and 2. You don't have to stick to that strictly, but essentially they want you to just be regular in your monitoring timing. Always do it at your same site and use consistent techniques. The methods are easy and inexpensive. And if you do it consistently, then you can come up with credible data. As always, if you don't think that you did it correctly, do the test again, as opposed to just guess that maybe you did it correctly. All right, how is bacteria counted? So the term is colony forming units, and it's often referred to by CFUs. And so that is a colony of bacteria cells that or it's bacteria that's grown into a small colony. And so the way that we identify that, each of those dots, whether they're blue or pink in that dish, that's an example of a Petri dish where we've collected a sample and then we've incubated it in an incubator and let it grow for a few days. And so it starts off looking like clear water. Each of those dots represents a colony of bacteria that's grown. And so we're going to count how many there were in that sample, and then extrapolate based on how many milliliters of a sample we took, how many are there per 100. So that's the typical standard, is colony forming units per 100 milliliters. Global Water Watch in their materials have a particular set of standards. They have this traffic light icon, and they say less than 200 is safe for frequent human contact. 200 to 600 is for occasional, and greater than 600 is unsafe. In Washington State, we have a stricter set of water quality standards. And so in our case, there used to be a little bit more complicated of a set of standards, but they've essentially simplified it and said, in Washington State, we want bacteria levels to be under 100 CFU per 100 milliliters. So there's a concept of listing a stream. We talked about this a little bit in the introduction class. There's a list, it's called this 303D list. It's a section of the Clean Water Act. And if somebody discovers through regular testing that a creek is impaired based on whatever water pollution characteristic, in this case bacteria, then it may get a listing on this 303D list as an impaired water body. If that's the case, then whoever's in charge of that water body, or at least each jurisdiction that affects that water body, will have to participate in a water quality improvement plan. And then they set maximum levels that they want to find when they do their tests. So they call that a total maximum daily limit. So for instance, in our case, we don't want to find greater than 100 CFU. So that might be the total maximum daily limit. And when they have this plan, they have to do regular testing to find out if what they're doing is having any effect on the bacteria levels in the creek.
Can I ask a question?
Sure.
So for that area of Thornton Creek where there was that high bacterial load, was that listed as a 303D, or is that an example of when that would happen?
That is an example of a creek that's listed, and so is Swamp Creek, actually, that we were doing our biomonitoring on.
Wow, there was a lot of those one level bugs for it being so contaminated. That's good.
Well, the bacteria that we find in the creek isn't necessarily an issue for like the insects that live in the stream or the fish. It's really more of an issue for like people in recreational contact with the creek.
Yeah, that makes sense. Okay, thanks.
Sure. Okay, the steps for bacteria monitoring. So the first thing we're going to do is collect the materials that we need. So there's these things that are called Petri dishes. They're these round plastic dishes, and they're coated with something in the bottom that after it sits for a while with a sample of water in it, it forms a gel. And the purpose of that is that if it just stayed as a liquid, it would all float around and you wouldn't be able to count colonies. So by forming it by causing a gel to form, it causes the colony to just grow in one fixed location so you can count it later. So Petri dishes is part of what we use. We also use what's called medium or media, and the particular brand or type that we're using is this technique called Coliscan Easy Gel. The medium is essentially like the food for the bacteria. So probably some form of sugar and water. That's what we will put our bacteria sample into when we collect it in the field. And then we'll pour that into the Petri dish so it can incubate. We use a thing called a pipette, which is like a little suction tube with a bulb on one end that you dip into the water and you can suck up a measured amount of sample. So that's how we collect a specific measured amount of sample. We use a thermometer just because we're checking our air and water temperatures. Usually we do this in conjunction with our water chemistry monitoring, so we don't have to double check, but that's another piece of equipment that we use. And then there's different types of incubators that you can use. The one that we're using right now is the one in the picture there, which is essentially an egg incubator for like raising poultry. We used to use some other incubators that were more improvised. Global Water Watch teaches you to do the absolute cheapest method. And so they have a model that you can follow of like a styrofoam incubator with some night lights in it that works. But we found it was a lot easier to use these because you can just set the temperature and not worry about it. So those are the items you want to get together. You want to have your data sheet. And another thing that we bring is we usually bring a little ice pack and like a little insulated container so that when we collect our sample, we can keep it on ice. So it essentially doesn't start reproducing already before we pour it out of the sample bottle. And you can do that for up to 3 hours. So we usually bring that. The alternative is to pour it into a dish on site, plate it on site. We typically don't do that because you'd be sitting there on site for a really long time waiting for it to gel, especially in our temperatures. So usually what we do is we collect it on ice and then bring it back to wherever it's going to incubate. So typically, like in my case, it's my house. Bring it back, plate it, and then put it into the incubator. Another part of getting your materials ready is you before you monitor, or at least before you plate, you're gonna label your Petri dishes and you're gonna set them up so that you can tape them shut. So what we do is you'll see a demonstration of this in a little video, but essentially you pour the sample in and then you either tape one side with just a little wrap of tape on one side, and the other side you make what we call a tab, which is essentially it's a piece of tape folded back on itself. So it still holds it shut, but you can untape it if you need to open it up. And the other thing you're going to do is label your samples. So let's say that we're doing 2 sites. We're going to do 3 what are called replicates for each site. So each time we go and monitor a given site, we're going to collect 3 samples. So if we were doing 2 sites, we'd want to label those so that we keep track of our 3 separate samples at each site. So you want to label those. So you've gotten your materials ready. You're ready to go out and do your sampling. Now you're going to go ahead and collect the samples at your field site location. So the first thing you're going to do is you're going to unwrap the pipette out of its package, and it's a sterile pipette. So you don't want to touch it on anything. Basically, you don't want to accidentally contaminate it and introduce some other bacteria source. You're going to collect your sample with the pipette, and that's usually anywhere. There's different size pipettes. We usually get either 1 or 3 milliliter pipettes, and we usually collect anywhere from a 1 to a 5 milliliter sample. Typically, usually 1 or 2 milliliters is a good size sample to collect. And when you're collecting your sample, you want to make sure that you're below the surface of the water. You're upstream from where you're collecting. So you're facing upstream if you're standing in the water. But you also don't want to be right down on the bottom, disturbing the bottom, because when you do that, you could generate more bacteria as well. So you'll collect your sample and you'll put it into a media bottle and then recap it and put it on ice to take it back to where you're going to plate it.
Hey, Eric.
Yeah.
Were you just saying that you put the pipette upstream of where you are, so whatever's on your boots, for instance, doesn't get in it?
Yeah, and mainly so you're not like stirring up the bottom.
Okay.
You could generate, if you're putting it downstream of yourself, just your movement and standing there, you could stir up the bottom, which could stir up more bacteria.
Got it. Thank you.
Sure. Okay, we're going to watch a short video on collecting samples.
Welcome to the Alabama Water Watch instructional video series. This video will show the proper method for collecting water samples used to determine the number of bacteria in the water body. This is the first step in a 3-step process. The second and third steps, plating the samples and determining results, are covered in their own separate videos. For each site being tested, you will need a cooler with ice, 3 media bottles at air temperature, a sterile pipette, and a thermometer. Label your media bottles by site and replicate. If you sample multiple sites, be sure to use separate data sheets or keep record of monitoring site and bottle numbers that are grouped in sets of 3 replicates. For example, if you have 2 sites, you can number the replicates at the first site as 1A, 1B, and 1C, and the replicates at the second site as 2A, 2B, and 2C. Upon arrival at the site, measure the air and water temperatures. For instruction on how to measure these temperatures, please watch the video called Water Chemistry, Air and Water Temperature. Once you are ready to sample, unwrap a sterile pipette from the bulb end and avoid touching the tip of the pipette with anything except the sample water. Fill the pipette with sample water. Be careful to not disturb the bottom of the stream. Carefully squeeze the bulb and expel excess water to the desired sample volume. 1 milliliter sample volume is recommended and is usually sufficient. Be sure to use the same volume for each of the 3 replicates and record volumes on data forms. Remove the cap from the media bottle and hold it without touching the inside. Squirt the sample water into the bottle of liquid media without placing your hand or face over the open bottle. Cap the bottle securely and place inside the cooler. Repeat these steps until all 3 media bottles are inoculated with water from the same spot. Keep in mind that media must be plated within 3 hours of collecting the sample and should remain on ice and out of direct sunlight at all times. Thanks for watching. If you have any comments or questions, give us a shout.
Okay, so that was collecting a sample. So the next thing that you do, sorry, I'm just going to check one thing. How did that video look to you guys? Did it look okay?
It was good.
Yeah, it looked fine.
Okay. So the next thing that we're going to do is what we call plating a sample. So essentially that's where you pour your sample into the Petri dish. So you want to have your incubator set up and ready to go. And the incubator temperature is 29 to 37 degrees Celsius. And one thing I didn't mention is that the media is usually stored frozen. So you got to thaw it out before you go out and do your sampling, or else you'll really be waiting there for a while. So it needs to be at room temperature as well. So you've collected your sample, you've come back where you're going to incubate it. Your incubator is ready to go. You're going to open the Petri dish enough to pour the media in. So essentially, you're taking precautions to make sure you don't introduce any other contamination. You'll just gently swirl it to cover the bottom uniformly. And then you'll place it directly in the incubator. And there's 2 ways you can do this. One is you can just pick it up and be careful and just kind of move it over into the incubator. The other way that you can do it, which I tend to do, is I actually often just put the plate in the incubator, pour my sample into the plate right there, and just put the lid on it right in the incubator, and then I don't even have to pick it up. So now we're going to watch a video on plating samples. Sorry, I don't think we have the right video queued up right there.
Plating and incubating. Now that we are back from collecting our samples, we are ready to plate and incubate our samples. The first step will be to pour the water samples that are mixed with the media into the pre-treated Petri dishes. For this test to work, the media and sample water must become a gel. For gelling to occur, the media must come into contact with the special coating that is found on the bottom part of the plate. Therefore, make sure.
Oops, sorry.
Sure that you pour the media into the bottom of the plate, which is slightly smaller than the top or the lid of the plate. Before plating, note the time. When you begin plating will be your incubation time on the data sheet. To plate, lift the top of the Petri dish just enough to pour the contents of a corresponding media bottle into the plate. Do not slosh the media onto the side or the lid of the plate. Close the plate. Gently swirl the dish in a circular motion on a flat surface to evenly distribute the media across the bottom of the Petri dish. This will help to reduce or eliminate bubbles that may appear in the sample. Slide the plate to the edge of the table and use another piece of tape to secure the untaped side of the Petri dish.
Hey Eric, the video is frozen.
Plate.
Oh, the video is frozen?
Yeah, audio came through, but the video is frozen. And it sounds like others are experiencing the same.
Okay, thanks for letting me know. Let me see what's going on here.
And there's no turtles.
Okay, let's try this again.
Plating and incubating. Now that we are back from collecting our samples, we are ready to plate and incubate our samples. The first step will be to pour the water samples that are mixed with the media into the pre-treated Petri dishes. For this test to work, the media and sample water must become a gel. For gelling to occur, the media must come into contact with the special coating that is found on the bottom part of the plate. Therefore, make sure that you pour the media into the bottom of the plate, which is slightly smaller than the top or the lid of the plate. Before plating, note the time. When you begin plating will be your incubation time on the data sheet. To plate, lift the top of the Petri dish just enough to pour the contents of its corresponding media bottle into the plate. Do not slosh the media onto the side or the lid of the plate. Close the plate. Gently swirl the dish in a circular motion on a flat surface to evenly distribute the media across the bottom of the Petri dish. This will help to reduce or eliminate bubbles that may appear in the sample. Slide the plate to the edge of the table and use another piece of tape to secure the untaped side of the Petri dish. Repeat these steps for each plate. You can immediately put your Petri dishes into the incubator in the same position they were plated, right side up. Check again to make sure the temperature in the incubator is between 29 and 37 degrees Celsius. Don't forget to fill in your incubation time, which began when you plated your samples. You should also note when your samples will be ready to count. This will be in 30 to 48 hours after they were plated.
All right, hopefully that video played better for everybody. And that's basically the process of plating. So they talked about incubation for at 29 to 37 degrees Celsius for 30 to 48 hours. One thing that is kind of an optional thing, they used to teach to flip the plates upside down if you have a larger sample. Sometimes condensation will form. I find that incubating at the higher end of this range with a 2 milliliter sample, I don't really have condensation issues, but people who say that they do have condensation issues will sometimes come back and check their sample after a few hours and then flip it upside down so that condensation doesn't form on the lid of the Petri dish, which could then drip down and somewhat dissolve the sample. So if you find that you're incubating and you have a problem with condensation, you can flip them. Otherwise, you don't have to. And then the final step is, now we've incubated it for our set time. Now we're going to interpret the results. So I'm going to show you guys a video on interpreting results.
Welcome to the Alabama Water Watch instructional video series. This video will show the proper method for determining the results of incubated water samples. This is the third step in a three-step process. The first and second steps, collecting the samples and plating the samples, are covered in their own separate videos. Remove plates from the incubator, being careful not to spill any liquid. Be sure to record the incubation period, which is optimally between 30 and 34 hours. When counting the number of colonies, it is helpful to use a white sheet of paper to split the plates into quadrants. Count all blue to purple colonies on each plate as E. coli. Do not count very small or pinpoint colonies smaller than the period at the end of a sentence. Count or estimate the number of all pink to dark red colonies on each plate as other coliforms. Estimates of more than 100 colonies should be reported as abundant, and estimates of more than 200 colonies should be reported as too numerous to count. This sample should be recorded as too numerous to count. If colonies appear as a ring around the edge of the plate or as one large blob, count as one colony. Record both the number of E. coli and other coliforms on the data form. The bacterial concentration in 100 milliliters will automatically be calculated in the Alabama Water Watch database. To calculate the bacterial concentration by hand, divide the number of E. coli colonies counted by the sample volume, then multiply by 100. Kill the bacteria by pouring about a teaspoon of undiluted bleach into the dish. Let stand for about 15 minutes, then dispose of with household garbage. Thanks for watching. If you have any comments or questions, give us a shout.
Okay, any questions on that? All right, so we're going to go through an exercise ourselves. So if this is a 1 milliliter sample, how many, when you guys throw out how many colonies that you count there in the plate that are blue?
7.
Yeah, that's what I get as well. Okay, so we have 7. So if our sample is 1 milliliter and we got 7 in the plate, how many would there be in 100 milliliters?
700.
700. Yeah, which is a lot. So then the other coliform on the plate, sometimes you can even section it down into like an even smaller chunk if you have a lot, so that you can count 1/8 and then just multiply them. So you count up how many you have in that section. And don't count the super tiny ones. And this will be kind of a slightly subjective gray area type thing. It's like, okay, what is that one? And the other thing is the colors aren't always perfect. So you have to do your best on this one, but you try to count the ones that are big enough that you can really consider them bigger than like a dot, like if you just dotted a pen or a pencil or a period at the end of a sentence. So in this case, we just counted 45 and 8 and 1/8. So doing the math, that would be 360 other coliform and 36,000 other coliform. And we really aren't super concerned with the other coliform, aside from, I think it gives kind of an overall sense of like, is your test working correctly? So if you only had E. coli and you didn't have other coliform, you might say, okay, my test isn't working correctly. So we count it, we enter it, but the one that we're really concerned about is the E. coli. So would this be water that you would want to drink?
No.
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 Would it be water that you'd want to swim in?
No.
No.
Okay, so we compare it to the results, even by Global Water Watch.
You can only have
standard.
like two.
What's that?
We're not muted.
I
That's
think he was
okay.
saying that for it to be safe, there would only be like 2 on the plate.
Yeah.
Maybe that's what you were saying. Okay.
Please muted us.
Yeah. So 700, it's greater than 600, it's unsafe for human contact. And in Washington state standard, you don't want to have it be less than 100. That's another reason actually why I tend to do a 2 milliliter sample these days, is because if you think about it, if you have a 1 milliliter sample and you count 1 colony, you can't count a half a colony. So you basically know whether you're either at or greater than 100, but you don't really know where you are if you're under 100. So that's why I tend to do a 2 milliliter sample. Okay, now let's say that we did a 5 milliliter sample. And how many E. coli do we have there?
7.
Yeah, still our 1, 2, 3, 4, 5, 6, 7. So in 100 milliliters, the math is a little bit more complicated, but basically we're going to divide by our sample volume and then multiply by 100. So that would make it 140. So it's not ideal by Washington state standards, but it still would be probably relatively safe for recreation. All right.
So
this,
sorry, I have a question. When you're
Sure.
doing sample sizes that are a larger milliliter, are the Petri dishes the same size? Like, do you use a bigger Petri dish?
no, the Petri dish is the same size, and the Petri dish will accommodate it'll accommodate a 5 milliliter sample. So that's not a problem. I guess the other thing I would say is just in terms of counting colonies, if you know that you have a site that tends to have a lot of colonies, you may want to edge on the smaller side of a sample size just to make it so that your counting is easier. If you know that you generally don't find that much, you might want to go with a bigger sample just because you're maybe more likely to find something, or you can be a little bit more accurate with it.
Okay. Yeah, because it seems like with a bigger sample, if there were a lot, they would be overlapping each other and more difficult to read. Is that ever happened?
Yeah, you can have samples where you have so many that it's just difficult to count, period.
Thanks.
Sure. So I think you guys get the idea of the different of counting, but that's basically the process of figuring out how many colonies you have, how many CFUs per 100 milliliter. So once you've gone through that, after you're done counting, you're going to record your results down in the bottom part. So going through this data sheet a little bit. So on all these data sheets, you're going to fill out who you're monitoring with, who's monitoring, the date, the time, location. Some of this information will be pre-printed if you're monitoring with us. You're going to look at the water body. Was there adequate depth? Was it, that's pretty much the only one that really pertains to us, is adequate, inadequate, or dry. You'll record air and water, your sample volume, the incubation time, which is the time that you start, that's your plating time, is when your incubation time starts. You'll record the temperature, how many hours. One item on here is the media expiration date. So the media does expire. So that's something you want to verify, is that it's still within date before you use it. And then plate it on site, yes or no. Typically we say no because we don't, but you can. And then transport it on ice for less than 3 hours, or you can transport it not on ice within 30 minutes. So you record all that. Now we've counted our colonies. We've got our results. We're going to record it on the sheet, and then we'll enter it into the database. And then final step is cleanup. And what Global Water Watch recommends and what they showed in the Alabama Water Watch video is to use some bleach to kill the colonies and then dispose of it.
Hey, Eric,
So yeah,
do you throw out the Petri dishes every time and then use new ones?
we use new Petri dishes for our samples. I don't always throw out the Petri dishes. Sometimes what I do is I clean the Petri dishes out and then use them again because they're useful for like that biomonitoring activity, for instance. But at a certain point, you're going to have more leftover Petri dishes than you know what to do with. So, but every time you do a new sample, you use brand new Petri dishes that come from a sealed pack.
And the coating comes with them.
Yeah, they're pre-coated.
Okay.
Yeah, we get both the Petri dishes and the media from a vendor called Micrology Laboratories. That's the one that developed this particular technique. And actually, at Auburn, the coordinator for Global Water Watch is researching right now into a different technique using, instead of Petri dishes, using a card, which it'll be a similar counting process, but it's like a pre-treated card that you would just put a sample on, and it would be maybe involve a little bit less materials and less waste than this technique does. Okay, now you've figured out what your bacteria level is. Now there's different ways that you can share that information. So think about maybe who you want to let know about it, what you'd like them to do with that information. Maybe see if that determines where you want to focus your monitoring. One thing I don't know if we talked about, I don't think we covered it at the beginning of this, but so you could monitor on one single site monthly. But another thing you can do is if you think you have a source that you're trying to track down, but you don't know where it is, you can also collect samples kind of all in one day or one short period along different points along the creek and compare those results. And that's a way that you can kind of see where maybe you have a higher source and help you track down where it's coming from. So this is an example of a Global Water Watch graph showing the different levels, and it's color coded, and they've actually changed it. They used to have the graph set up for Global Water Watch standards, and they've actually changed the color so that it shows basically either red or green, depending on if it's over or under 100. So that's one thing you can do, is use that. You can also take pictures of your samples and make them into a poster. This is something that Lake Forest Park stream keepers have done, and that they're a group that we partner with that monitors streams in Lake Forest Park. And you can see their poster illustrates how on the sites that they monitor after the first heavy rain, that you can see that they had a lot more.
Of shit.
This is an example of where they did a bunch of simultaneous data collection and kind of identified where the hot spots were. You can also, depending on where you're sampling, this was a place at Auburn University where they found super high colonies. And so they ended up basically putting up a public advisory sign saying, hey, super high contamination.
Behind a frat house.
Yeah, don't want to get in this water.
It was behind a frat house.
Quite possibly.
Is it, I guess that previous slide would help inform, but so once the input of the bacteria is identified and eliminated, how long typically can those bacteria persist? Or is it typical that they're just like dispersed?
That's a good question. I think because we're sampling a moving water body, unless you're sampling a fixed water body, you're kind of getting what's coming downstream. So it's more like what's continuously in that water body is what you're detecting. So I don't know how long they would live, like individual bacteria in a water sample, but that's a good question. Here's another example of how people use the data. So they brought their sampling sheets, they drew a big map of their watershed, and then they converted that to a map showing the watershed and where the bacteria levels were higher. Another way that this data is used, there's some water keeper groups that contribute to something called a swim guide, and they test water at different beaches for bacteria levels. Around here, mostly, like if you're in King County, I think King County Public Health tests swimming beaches in particular, but there might be a place like, for instance, over at Wallace Swamp Creek Park. I don't know that they test or post anything there. So that might be a place where if we found really high levels, we might talk to the city and say, hey, probably want to post an advisory sign here. And here's a poster from Global Water Watch Mexico, just kind of sharing what they found and what it means. And that is it. So these are some of the organizations that helped us get our program going. So thanks to all of them, and thanks to you guys for participating in our class. So I'm going to stop the recording now, and then I will answer any questions about our upcoming practical session on Saturday.